via phosphodiester. Okay, what did it say? Got it. Never mind. It told me the recording was started. Okay. Anyway, so deoxyribonucleotides, which are all linked to each other via phosphodiester bonds, right? So when we say these are all linked together, these are two strands we're talking about. So you have two strands running together like this, and they actually kind of coil around each other like that. And the structure is called a double helix structure. Very characteristic of DNA. Whenever you hear the term double helix, always think of a double strand of DNA wrapped around like this, okay? Now, these two strands are complementary to each other, and they run anti-parallel. What do I mean by these terms? Complementary and anti-parallel. I'm just going to skip forward a few slides. Coming to this slide here. So we said DNA is a double-stranded structure, two strands, right? So I want you guys to look at this strand here. Sorry, this part of this image here. Notice that there are two strands, and each of them seems to have these like pairings going on. There's like these pairs of nitrogenous bases that are forming. So what happens is the nucleotides of DNA form pairs from across, like basically across the DNA structure. Each strand has nucleotides which form a pair with a nucleotide on the opposite strand. And it's always the same nucleotides forming these pairs. And these are called base pairs. So on the first strand of your DNA, you're going to have guanine, you're going to have adenine, you're going to have cytosine, and another one would be thymine. And each of these has a specific nucleotide which they bond with or which they pair with. So guanine, G, for example, will always pair with cytosine. So that's C. G and C are always going to be together throughout the entire structure of the DNA. Then you have your A, which is adenine. A is always paired with T, which is thymine. A and T are always going to be together throughout the structure of your DNA molecule. It's always going to be, it's always going to be A and T or T and A, and it's always going to be C and G or G and C. Like it's either way, it works, okay? So adenine and thymine is one pair, and then cytosine and guanine is your second pair. And this is very important to remember. Now, that's what I mean when I say these strands are complementary to each other. The first strand complements the second strand because each of the bases on the first strand, they match each of the bases on the second strand. They form pairs, and that's why they're called complementary strands, because they complement each other. Now, coming to the fact that they're anti-parallel, what does that mean? Now, looking at this image here, you'll notice that the first strand on this side is going from the 5' all the way to the 3'. It's going upwards like this, right? And the opposite strand is going from 5' at the top to 3' at the bottom. It's running in the opposite direction. So the first one's going up and the second one is going down. And that's what I mean by anti-parallel. So there are two strands, they complement each other, and they run in opposite directions. The first one goes from 5' to 3' upwards, and the second one goes from 5' to 3' downwards. Now, these two concepts are really important to keep in mind. The fact that they complement each other and the fact that they are anti-parallel, okay? Now let's go back to the first slide here. All right, so we covered DNA. Now let's come to RNA. RNA stands for ribonucleic acid. In this case, it is a single strand. It's not a double strand, it's a single strand, and it is made up of ribonucleotides. That's the first difference. It's a single strand. The second difference, it's made up of ribonucleotides instead of deoxyribonucleotides. We'll come to the structural differences between these two in a second. But for now, what I want you to know is RNA is a single strand and it's made up of ribonucleotides. DNA is a double strand made up of deoxyribonucleotides. Okay, another thing is the fact that RNA can have many different functions, whereas the DNA serves a single purpose, which is to carry the genetic information of the cell. So what does DNA do exactly? The DNA contains all the genetic information which encodes everything to do with the cell or the organism. Every single thing that the cell does, every instruction that is carried out by the cell or by the organism, everything, the entire genetic makeup of the cell or the organism is all encoded within the DNA. And this is known as the genotype. The genetic makeup of an organism or the genetic makeup of a cell is known as its genotype. And all of this genotype, this entire genotype that makes up the cell or the organism, is all encoded within these two strands of the DNA molecule. Okay, so far so good? Any questions, guys? All right, so DNA and RNA, we said that they're both made up of nucleotides. The difference is in what kind of nucleotides we're talking about, right? So in DNA, we are talking about deoxyribonucleotides. And in RNA, we're talking about ribonucleotides. Now, what does that mean? What is a nucleotide, first of all? So a nucleotide is a structure. It is that little unit that makes up the DNA or the RNA. And that unit itself is made up of three things. You're going to have a nitrogenous base, you're going to have a sugar, and then you have a phosphate group. And this is what that looks like. So this is your pentose sugar. It can be a ribose pentose sugar if you're talking about RNA, or it could be a deoxyribose sugar if you're talking about DNA. And then regardless of whether it's an RNA or DNA, there's always going to be a phosphate group, and then you're going to have a nitrogenous base. Sometimes you might have more phosphate groups attached here. Sometimes you might have two phosphate groups. Sometimes you could have a total of 3 phosphate groups attached here. But the point is the same. There's always going to be phosphate, there's always going to be nitrogenous base, and there's always going to be a sugar in the middle. Okay? Now that's the first difference between RNA and DNA is the sugar itself, whether it's a ribose or a deoxyribose. The second difference is actually the nitrogenous bases themselves. They are not the same bases in DNA and RNA. So in DNA, you have 4 bases. You have adenine, thymine, cytosine, and guanine. Now in RNA, they're the same except for one difference. Instead of having a thymine for a nitrogenous base, it is replaced with a uracil. Remember how earlier I told you that there's always pairs that are forming in DNA? Adenine is always with thymine. Wherever there's an adenine, you can expect it to be paired with a thymine. Now in RNA, it's the uracil that is being paired with the adenine. So wherever you see an adenine, if it's a double-stranded structure, you can just assume that's going to be a uracil that will be produced or that will be paired with the adenine on the other end. So it's exactly the same. The only difference is that thymine is now replaced by uracil. Now, what do these nucleotides actually look like in terms of structure? This is what they look like. Okay, so this is your nitrogenous base. This is your phosphate group, and this is your pentose sugar in the middle. Okay, now deoxyribose. Look at this H over here. It's a single sole hydrogen, and that's why it's called a deoxy, because the oxygen is just gone. But then when you look at the ribose, there is an OH. It's a whole hydroxyl group. And this is why this is called the ribose pentose sugar. So in this case, in case of RNA, it's a ribose. And in case of DNA, it is a deoxyribose. Deoxy meaning the oxygen has just been swiped away, just been taken out, knocked out. Okay? And that's the first difference. Second difference is in the thymine versus uracil. So in DNA, you have thymine, and this is the structure of thymine. I don't think you need to know what it looks like exactly. You might, I'm not sure, that depends on your professors. But in case you need to know, just focus here, and inshallah, you'll get the hang of it. The only difference between thymine and uracil is the fact that thymine has this methyl group, and uracil doesn't have that methyl group. And that's the primary difference between, the nitrogenous bases in the DNA versus the nitrogenous bases in RNA. This is again an image which just shows you the difference: thymine versus uracil, deoxyribose versus ribose, and yeah. Okay, so we said earlier that all of these nucleotides are connected with phosphodiester bonds. What is a phosphodiester bond? So a phosphodiester bond is what is formed when the phosphate group of one nucleotide bonds with the hydroxyl group from the sugar of a different nucleotide. So the hydroxyl group of this one binds to the phosphate group of this one. The hydroxyl group of this one binds to the phosphate group of the next. The hydroxyl group of that one binds to the phosphate group of the next. And it's always phosphate binding with hydroxyl, phosphate binding with hydroxyl, phosphate binding with hydroxyl. And these form the phosphodiester bonds, which will then link your nucleotide. And this is what that looks like. This is the phosphodiester bond that we're talking about. So this here, this green nucleotide, is all one nucleotide. And this phosphate is part of this nucleotide. And you'll see that the top of this phosphate group is bound, or has bonded with the hydroxyl group of the pentose sugar from this nucleotide. Same thing here. The hydroxyl group has bonded with the phosphate group. Again, this hydroxyl group would bond with the phosphate group of the upcoming nucleotide. Is that clear? Do you guys understand phosphodiester bonds? Do you guys understand deoxy versus ribose? Do you guys understand the fact that RNA is always a single strand, DNA is always a double strand? Everything cool so far? Yep, perfect. Okay. Now coming to the same slide that I went to earlier. This slide has been covered, but just so you guys just picture it together in your head, the sugar, phosphate, and base, together they form a nucleotide. This is a single strand. Then a DNA has two strands which are running in opposite directions. And finally, they form something called a double helix structure, where they are tightly wound around each other. And each of them has base pairs. T is always with A, and C is always with G. Now coming to RNA. RNA is always a single strand. And once again, the difference is in the fact that it's uracil, not thymine. And this is what it looks like. It looks like a DNA strand, but you just kind of pull it apart the second one. And so it's a single lonely strand on its own. And yeah, this is a table which kind of just summarizes everything that I've said. You guys can go over it later just to make sure you understand everything. But one point that I didn't mention earlier is the fact that DNA is very long, because DNA encodes the entire genotype, the entire genetic makeup of the cell is all encoded within the DNA. So you can imagine it's going to be very, very long. But when it comes to RNA, RNA is like taking a piece of the DNA and transcribing it into an RNA. So it's only representing a portion of your DNA. Therefore, RNA is usually very short. It only represents a certain gene, maybe at most 2 genes. And it's usually pretty short in comparison to the very, very long DNA. The DNA is going to consist of many, many, many genes. The RNA is like a representation of one of those genes. RNA is pretty short. Okay. Now coming to the final few definitions that we need before we get into the process of transcription. So what I was mentioning earlier, just a few minutes ago, is actually gene expression. So gene expression is the conversion of information stored in that very, very long DNA molecule into something which the cell is now going to use. So let's say the DNA molecule encodes for a certain protein, and that protein is needed by the cell, right? But for the time being, it's only represented as a gene in the DNA. What happens is that gene is then expressed through transcription and through translation, and then it produces a protein. The end product is a protein, which then goes out into the cell and performs its function. So that one gene in the DNA that coded for that protein then undergoes transcription and translation and becomes that protein. So that protein is now existing. Before it didn't exist. Before it was just a representation. It was coded as a gene that forms a protein, an actual molecule, or an RNA or something, a functional molecule that can now be used by the cell. And this is termed as gene expression. And gene expression occurs in 2 stages. The first stage is transcription, where your DNA is copied into RNA. And this is done by something called the RNA polymerase. That's the enzyme that converts the DNA into RNA. Then you have your translation, which is when your RNA is converted into the actual protein. So it goes like this. The gene or the coding itself is in the DNA. the DNA forms the mRNA or the RNA, and then that forms the actual protein, which is then used in the cell, or goes out and does whatever. And that's a whole process. Translation is done by ribosomes. I think that's a different lecture. Not sure if it's in your midterm tomorrow, but ribosomes are something different. We're not going to be covering that today. Just for the sake of your information, translation is done by ribosomes. They are the ones which produce the protein. Okay, so through gene expression, what's happening? Your genotype, which is stored in your DNA molecule, is expressed and converted into phenotype, which is the actual protein or the physical representation of whatever was coded in the DNA. Now, let me explain it to you in this way. Let's say a person has a gene that codes for blue eyes, okay? So that's a gene that's located somewhere in their DNA, and that's part of their genotype. It's part of their genetic makeup. Now, when that gene is transcribed or it undergoes transcription, it forms mRNA, and then it forms a protein, which then goes and deposits itself in the person's eye. It forms a blue protein, let's say. Now, when I look at that person, I see their blue eyes, and that is the phenotype. That is the physical representation of the gene encoding blue eyes that was in their genotype. Does that make sense? So gene expression is essentially the conversion of genotype, which was stored in your DNA molecule, into phenotypes, which are observable physical characteristics of an organism. It could be blue eyes, it could be, I don't know, brown hair, it could be blonde hair, it could be like skin color, whatever. It doesn't even have to be a physical trait. It could be whatever, it could be like the shape of a person's cells. It could be something, anything, But what I mean is genotype is converted into phenotype, and this process is gene expression. It has two steps. The first is transcription, done by RNA polymerase, and the second is translation, done by ribosome. Okay, and this is a slide which talks about different kinds of RNA. The most important one is the one that I've highlighted, which is the messenger mRNA. Messenger mRNA is the term used to describe the RNA that is produced after the first transcription from the DNA. So you have your DNA, it undergoes transcription by the RNA polymerase to form the mRNA, which then goes to the ribosome to form the protein. Okay, so for this lecture, what you need to know, the most important one is mRNA. However, there's different kinds of RNA. Remember, RNA is really short, it's always really short. So there's different kinds. And you could have transfer RNA, you could have ribosomal RNA, you could have all of these. You can read those, at your own time. For this lecture, you just need to know messenger RNA, shortened as mRNA. What does it do? Transfers the code from the DNA into some form which is digestible or which is understandable by the ribosome, which will then produce proteins through translation. All right, we're done with the initial part of the lecture. Half the lecture is done. If you guys are still with me, you guys are doing great. All right, so coming to transcription. One of the most important enzymes in transcription, like we mentioned earlier, is RNA polymerase 2. There's different types of RNA polymerase. There's RNA polymerase 1, there's RNA polymerase 3. But for this lecture, the most important one overall, not even for this lecture, overall, the most important one is RNA polymerase 2. It produces the majority of, or it's responsible for the transcription of the majority of proteins in the body. So RNA polymerase 2 is the star for this lecture, okay? So what this enzyme does is it synthesizes a new RNA molecule based on the genes encoded within the DNA. Now there's two important things that we need to know about RNA polymerase 2 in eukaryotes. The first thing is that RNA polymerase 2 is inhibited by a certain toxin called alpha-amanitin. So alpha-amanitin is found in mushrooms, and usually the exam questions that they ask you are going to be something like, oh, well, there was this family, they were on a picnic, and they decided to try these wild mushrooms that they saw growing in the corner, and they were like, why don't we try those? And then like half an hour later, entire family was in the ER and they had nausea, vomiting, diarrhea, cramps, overall not doing well, pretty sick. Why? Because those mushrooms contain alpha-amanitin. And this is like a very common question. Examiners love asking this. And they will always ask you, oh, which enzyme was inhibited by the mushroom, by that ingredient in the mushroom? It's always RNA polymerase 2. Okay, that's the first thing. Second thing is that RNA polymerase 2 does not proofread. It has no mechanism for proofreading. You know how DNA replication has proofreading? RNA polymerase doesn't do that. It's kind of like it's not bothered, just couldn't care less. But what that means is that RNA polymerase has a tendency to make errors and mistakes. And that's a bad thing, because that means that if it's not careful, it could result in the production of proteins which are, not useful to the cell, accumulation of the wrong kind of protein. It could be really bad. But that's too deep for you guys. All you need to know is that it doesn't have a proofreading mechanism, which means that it's kind of just writing without actually reading when it's writing. It can make mistakes. Okay. Now, the process of transcription itself. How does transcription start? You have your RNA polymerase. What it does, it looks at your DNA and it assesses the DNA for a certain gene portion. It is looking for something called the promoter region. And this is your promoter region here, okay? And the transcription starts when the RNA polymerase recognizes this promoter region. So it looks at the entire DNA, or it looks at segments of DNA, and it's looking for this promoter region. This promoter region is just a region of nitrogenous bases or nucleotides a little bit ahead of the point where transcription is supposed to start. So it's a little bit ahead of that point. And on this promoter region, you will have other factors binding to it called transcription factors. We're going to come to transcription factors in a bit. But essentially, the transcription factors bind to the promoter region. And once the transcription factors are there, that is what helps the RNA polymerase to recognize the promoter region. And it binds the promoter region, and then it starts its transcription process. Okay? So the RNA polymerase recognizes and binds the transcription start site or initiation site where the promoter region is, and then it proceeds with transcription. Now, how does it proceed with transcription? What does it do? Now, what it does is it pulls apart the two strands of DNA. So the DNA was coiled like this, right? It pulls apart the two strands because it can only focus on one strand at a time. When it replicates or when it forms the RNA, because it's going to produce RNA, it can only form it from one strand at a time. So it can only read one strand. So it has to split the two strands. It forms a single-stranded template. This is your two strands of DNA. This bottom strand is the one that the RNA polymerase is going to read. And this one is called the single-stranded template. Now, I know this slide is a bit crowded. Stick with me, okay? So for any gene, the RNA polymerase is only able to read that lower segment, that single strand of DNA. And that's called the template strand. So what happens is the RNA polymerase is going to go this way. Imagine it is like eating the template strand. It's going to move from left to right. So it's moving from the 3' end of the template strand towards the 5' end. So look at the bottom strand here. This is a point where a lot of people get confused. So I want to make sure that everyone is good with this before we move on. Look at the bottom strand here, okay? This is your 3' end of your template strand, right? 3' end. And this is the 5' end. Now, if your RNA polymerase is binding here, and if it's moving in that direction, it's moving that way, it is reading the template strand starting from the 3' end towards the 5' end, right? So RNA polymerase reads the template strand from the 3' end towards the 5' end. And this point is very important to remember. So the polymerase is reading it from 3' to 5'. What about the strand that it's producing? Because ultimately the RNA polymerase is reading the template strand and it is adding nucleotides to its own strand that it's forming. As it goes along, it keeps reading and it keeps adding, right? So when it's adding, what order is it adding in? It is adding in the 5' to 3' direction. Because if it's reading in the 3' to 5', the only way it can add is at the back end of the strand that it's producing. So the strand it's producing is coming off of this side, right? It's kind of going down this way. It's falling off, right? As you can tell in this image. And the 5' end is the one that it produces first. And as it goes along, the RNA polymerase is adding nucleotides to the 3' end of this brand new mRNA strand that it is producing. If that's a little bit hard to understand, come look at this diagram on the right. So this is your template strand, right? Template strand is running from the 3' end to the 5' end, and the RNA polymerase is reading it in this way, from the 3' end to the 5' end. Now, when it adds nucleotides, because that's the whole purpose of the RNA polymerase, it has to form RNA that is complementary to the template strand. As it adds nucleotides, it is simply adding in the direction of 5' to 3'. And another important point to remember is that RNA doesn't have a thymine. So here, where you have an adenine, you're going to get a uracil on your RNA. Where you have a cytosine, you're going to get a guanine, cytosine, guanine, and where you have an adenine again, you're going to get a uracil. And it keeps adding to the 3' end. So when the new RNA strand is formed, it is produced in the 5' to 3' direction. But when it is reading, it's reading in the 3' to 5'. What is the polarity of the phosphodiester bond? I don't know. I actually don't know. I don't know. I can look it up for you if you want. But for the time being, I don't think you need to know. However, I will get back to you, inshallah. Okay. Okay, now coming to an important differentiation, distinguishing the template strand from the coding strand. When I was in first year, I used to have a lot of difficulty understanding why the coding strand was called the coding strand, because I was like, the RNA polymerase is not reading that strand, because that strand is up, so why is it even called the coding strand? But the truth is, it's called the coding strand because it is a replica of the RNA, the new RNA strand that is being produced. So here's your template strand, right? And the top one is your coding strand. Your template strand is the template which forms the mRNA. So the mRNA that is produced is complementary to your template strand. Each of the nucleotides on the template strand has a complementary base or has a complementary nucleotide on the new RNA that is being produced, right? So it is called that's what's called the template strand. So the mRNA that is produced does not match the template strand. It's complementary, but it's not identical. However, the coding strand is the strand which is in fact identical to the RNA that is produced. How come? Let me just open my pen here for a second. All right, so if you look at this one here, the template strand is going to have a certain nucleotide here. And that nucleotide has a matching base pair on this side, on the RNA, right? Not the best drawer, so just gonna stick with me. However, the coding strand has an exactly the same nucleotide on this end, because ultimately it was the coding strand that was matched with the template strand, right? They were together, they were both matching base pairs, they were, paired up. So when the coding strand is separated, the RNA strand that is formed is actually a replica of the coding strand, which was initially paired with the template strand. And that's why it's called the coding strand, because the RNA strand that's produced is the same as the coding strand. The only difference being that if in the coding strand there was a thymine, in your RNA, it's now going to be called a uracil. It's not called a thymine anymore, it's called a uracil, right? So this bottom image here shows it really well. This is your template strand. Wherever you have an adenine, it becomes a uracil. Whereas on the coding strand, it would be thymine. But apart from that, the coding strand is exactly the same as the RNA, where it's an A, it's an A, G is the same, A is the same, C is also the same. The only difference is the fact that thymine is no longer thymine and it is uracil in the RNA. So it's exactly the same. It corresponds to the same sequence. The coding strand corresponds to the same sequence as the mRNA, which was built from the template strand. Any questions before I move on? Is RNA polymerase the same enzyme that unzips the DNA? Yes, in this case. RNA polymerase is actually made up of a whole bunch of things. For the sake of your information right now, all you need to know is that RNA polymerase is the one that kind of splits it apart and then it reads. It's not the same as the one which is used in DNA replication. DNA replication is a separate process. But in this case, RNA polymerase is the enzyme that unzips it. There's another person asking, okay, I don't think that's a question. Yeah, okay. All right, any questions so far? Is there anything that you guys don't understand? We are almost done with the important parts of this lecture. Okay, perfect. All right, so how does the RNA polymerase know where to start and stop? So RNA polymerase, as I said earlier, it recognizes the promoter region, right? And it also recognizes something called the terminator region or the stop region. And that's where it knows how to end. So the RNA polymerase reads the entire DNA segment from the promoter region to the terminator region, and that's where it knows how to stop. And when it's done reading, it produces an mRNA called the pre-mRNA. Because what happens is this mRNA actually needs to undergo further processing before it can become what is called the mature RNA. But for now, all you need to know is after this whole transcription process is done, it is called the pre-mRNA. Okay, now for a couple of questions. This question was in your lecture, so you might get it, you may not. I hope you guys get it though. But before we answer the question, one thing that you should know, whenever we write a base sequence, whenever we write it out by convention or by a standardized way, that whenever we write out a base sequence, it's always written in the 5' to 3' direction, no matter what it is like in the actual DNA itself. When we write it out, we always write it in the 5' to 3' direction. So during RNA synthesis, the DNA template sequence, TAGC, would be transcribed to produce which of the following RNA sequences? Do you guys know what the answer might be? People are saying E, a couple of people saying E, a whole bunch of people saying E, and you guys are correct. Now, why is it E? Because remember, this is written in the 5' to 3' direction. However, when the RNA polymerase is going to read it, it's going to read it in the 3' to 5' direction. Remember? So it's going to read it starting from C. It's going to read it as CGAT. And when it adds nucleotides, it's going to add a nucleotide for C first. That's going to be G. Then it's going to go to G. It's going to add a C. Then for A, it will add a uracil. And then for T, it will add an adenine. Sorry, adenosine. Keep saying adenine. Adenosine. Is that clear? Does everyone understand why it's E? Personally, this used to be something that used to confuse me a lot. I didn't get it, but live and you learn. All right, one more question, just to make sure you guys understand this, because it's very likely that a similar question to this will be in your midterm. Another question. So during RNA synthesis, the DNA template sequence AACT would be transcribed to produce which of the following RNA sequences? Which one would it be? I see people saying C. More people saying C. You guys are correct. MashaAllah, you guys are getting it. Good to know. All right, so the answer is C. Why is it C? Because the RNA polymerase is going to be reading it from the TCAA. So tree translates to A, which is the opposite pair. C, the pair is G. And then for A, it's going to be a uracil. So 2 uracils added to the end, AGUU. Now I have one more question, but this one is a little different. So I want you guys to read it carefully. During RNA synthesis, the RNA strand produced from a certain DNA template sequence is UGCA. Which DNA template would be used to transcribe, or would be transcribed to produce this RNA strand? So here I'm giving you the RNA. I'm not giving you the DNA. I'm giving you the RNA. What DNA would produce this? I see a bunch of people saying D. People are saying D. And you guys are right. I'm actually so happy. Oh my God. Okay, you guys are right. Do you guys understand why? Because once again, you have to sort of work backwards in this case and be like, okay, well, you know, the DNA template is going to be read in the 3' to 5', but the one that's produced is in the 5' to 3' direction. So you have to reverse this, reverse it in order to get the DNA template that would have produced this this RNA strand. Is that clear? Is everyone following so far? This is probably the hardest question you could get for this concept specifically when it comes to, you know, the reversing stuff. This is probably the hardest thing that you could get. All right. So now coming to the very end of this lecture, just a few other things that we wanted to talk about. We're going to talk about the promoter region in a little bit more detail. So the promoter regions are specific DNA sequences that are located upstream of the area where transcription is going to start. And they contain certain sequences like the AT-rich sequences known as the TATA box or the TATA box, and the CAT box or CAAT box. They're known as AT-rich sequences because they contain a lot of adenosine and a lot of thymine, like a lot of T's and A's. So when you look at this region here, within the promoter region, the TATA box, you'll see a lot of T's, a lot of A's, because it's essentially just an AT-rich sequence. That's what that means. Now, the promoter region is really a binding site for the transcription factors. The transcription factors must first bind to the promoter site before the RNA polymerase can recognize the promoter region. In order for the RNA polymerase to recognize where the promoter region is, it has to have transcription factors bound to it. So this is what happens. This is your promoter region here. Transcription factors bind to it. More transcription factors bind to it. There are loads. And then finally, the RNA polymerase realizes, oh, there's a whole bunch of transcription factors bound here. They've all just come and collected. Maybe this is where the promoter region is. And that's how it recognizes the part of the DNA where the promoter region is present. Now, what are transcription factors? They are basically helper proteins that allow the RNA polymerase to recognize where to start transcribing from. They bind to the DNA on a promoter region, and they help the RNA polymerase essentially just sort of realize where a promoter region is. Another thing they do is they combine with the RNA polymerase to form a complex called the pre-initiation complex. You don't need to know much about this pre-initiation complex. All you need to know is it's sort of a complex which helps the transcription process. It helps initiate the transcription process as soon as new nucleotides are available for the RNA polymerase to add to its RNA strand. And it's just called the pre-initiation complex. So if someone asks you what is the pre-initiation complex made up of, you're going to say it's made up of the RNA polymerase, typically RNA polymerase 2, along with all the transcription factors which were present at the promoter gene. What do the transcription factors do? they help the polymerase recognize which part of the gene is the promoter region. Okay. All right, coming to the final thing, enhancers. What are enhancers? So enhancers are like certain parts of your gene, certain parts of the DNA, where if a transcription factor were to bind to this small portion, the rate of transcription is greatly increased. The expression of that gene is enhanced. So a certain enhancer can be present somewhere near the promoter region. And if a transcription factor binds to it, that whole gene that is represented by that sequence of DNA is going to be enhanced. It's going to be transcribed by a lot more than it would usually. So what they do is they just increase the activity of that promoter region. They're kind of like a catalyst. They stimulate more and more transcription of that gene. And they have a stimulatory action over a very long chain of genes. It could be a huge gene, and that one enhancer could increase transcription of that gene by like tens of thousands of times if necessary. So there's different kinds of enhancers, but for your understanding and just for your, just to help bring everything together, all you need to know is that they're little short DNA sequences, and what they do is they increase the transcription rate of a gene. So that same chromosome, that same segment of DNA is now going to be expressed a lot more because of that little enhancer that was present. Okay. Now the final part of this lecture, if you guys are still here, congratulations. Prokaryotic transcription. So the genome in prokaryotes is, number one, much smaller than eukaryotes. What are prokaryotes? Single cell, like bacteria, right? So the genome is much smaller. And second, there is no nucleus. So they would just be floating in the cytosol. Even when there is a nucleus, typically the genome or the DNA of the bacteria floats around in the cytosol. And it's usually single-stranded, not double-stranded. It can be double-stranded, but it's just loosely floating around, which means that it's much more accessible for the polymerase of the prokaryotes, and it's much easier for the prokaryotic polymerase to perform transcription. And one more thing, there's only one polymerase in prokaryotes that produces all the kinds of proteins, all the kinds of RNA in the prokaryote. It's not different kinds, it's one single one which produces all types. So mRNA, hnRNA, whatever kind of RNA that you need in the prokaryotes, all that same polymerase that is doing all that. One point to note, the prokaryotic RNA polymerase is inhibited by rifampin, which is a drug used to treat tuberculosis infections. This is a point that the professor mentioned, so I'm thinking it's of importance for you guys. But essentially, tuberculosis is caused by what? It's caused by a bacteria called Mycobacterium tuberculosis, right? A bacteria. So when you give a person with a tuberculosis infection, when you give them rifampin, rifampin will then inhibit the RNA polymerase within the bacteria. It's going to stop the bacteria from reproducing or from performing its cellular function. And that is how you kill the bacteria off and you treat the infection. Okay. This is probably like one of the last few slides. In prokaryotic transcription, you need a couple of things. Instead of having promoters or enhancers or transcription factors, what you need is something called a sigma factor. A sigma factor is kind of like the promoter region for a prokaryote, and it is, as soon as the initiation occurs, sigma factor is released. And then finally, at the termination, you have something called a rho factor here. And that is how the RNA polymerase in prokaryotes knows where to let go. So sigma and rho, simple as that. Final slide, prokaryotic versus eukaryotic polymerases. In prokaryotes, is a single RNA polymerase. It's called alpha-2 beta prime, I think, beta prime, yeah. In eukaryotes, you have different kinds of RNA polymerases, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and each of them produces different kinds of RNA. And then in prokaryotes, you have a sigma to initiate the promoter. In eukaryotes, you have transcription factors, which bind to the promoter region so the RNA polymerase can recognize them. In prokaryotes, you need a row to terminate. In eukaryotes, you don't need a row, but sometimes there might be a termination signal or a termination point for the RNA polymerase to recognize where it stops transcribing. And finally, prokaryotic polymerases are inhibited by rifampin, and eukaryotic polymerases are inhibited by alpha amanitin, which is in mushrooms. And that is it. That's it. Thank you guys for listening. I hope you guys understood. Do you guys have any questions? Any questions? I would be happy to answer. Good luck in your midterm. I hope you guys do really, really, really well, inshallah. Inshallah, it'll be easy. Make sure you do the past exams. Focus on those. Make sure you review questions. Someone's asking, what is the difference between RNA polymerase 1, 2, and 3? So the different kinds of RNA polymerase, each of them is specific for a certain kind of RNA. However, for the majority of proteins that are transcribed and translated from DNA, it's always going to be RNA polymerase 2. So it's always going to be RNA polymerase 2 that produces the majority of proteins necessary for a cell. However, just for your knowledge, for the sake of your knowledge, you should know that there are other kinds of RNA polymerases. Number one, RNA polymerase number 1 and RNA polymerase number 3. And this is what they produce. It's on this slide here. So RNA polymerase 1 produces rRNA. RNA polymerase 2 is mRNA, produces mRNA, which is like the biggest, the most important one. And RNA polymerase 3 produces transfer RNA and other kinds of RNA. All right, someone else is asking, can I please repeat enhancers? Okay, where's enhancers? All right, so enhancers are essentially just little sequences of DNA, of segments of DNA, which are present ahead of the promoter gene, a little bit ahead of the promoter region, sometimes present on the promoter region. And what they do, if they're present, they're not always there, but if an enhancer is present, and if a transcription factor happens to land on that enhancer, once that transcription factor is there, the RNA polymerase, which looks out for the transcription factors, is going to recognize that enhancer and is going to bind to the enhancer part of that promoter region, right? Now, once it binds, the enhancer is kind of like a catalyst. It's like adding fire to the reaction, you know, adding a spark. What happens is, because of that enhancer, that transcription is multiplied, and that segment of gene, which only had the promoter region for it first, that segment of gene is now going to be transcribed multiple times over. So the rate of transcription of that gene is going to be enhanced. It's going to be increased. It's basically an enhancer is kind of like giving a boost to the promoter region. Promoter region itself was essentially just a guide for the RNA polymerase to know where to start transcription, right? And how is the RNA polymerase recognizing the promoter region? Because of the transcription factors which were bound to it, right? In case of enhancers, if an enhancer is present on the promoter region and a transcription factor is bound to it, the RNA polymerase is going to see that and is going to sort of like attach to it. And that will cause an increased activity. Whatever the activity of the promoter was initially, the enhancer is going to double, triple that, multiply that by many times. So it's kind of like a stimulant. That's really just all it is, an enhancer. Is that clear? Okay, so someone is saying, can you explain monocistronic and polycistronic? So this is not in my presentation, but it was in your other presentations. Let me go back to it. Where's the comment? Can you guys see the second presentation? I'm not sure. Hold on. I think you can't. Okay, here. I think you guys can see it now. All right. So someone is asking, can you explain the difference between monocistronic and polycistronic? So monocistronic is essentially just a transcription from a single gene that encodes for one single protein. And this is typically how it's done in eukaryotes. Any mRNA that is produced, like we said, the DNA is very long and it consists of many, many, many genes. The RNA that is produced from that one segment of DNA is going to represent a single gene. That's what that means. Monocistronic. Cistron is another name for gene. So monocistronic means single gene. It represents a single gene. However, in bacteria, one mRNA which is produced can represent multiple genes. It can encode for multiple at a time. It's not like eukaryotes. And that is why it's called polycistronic. So one mRNA can be a polycistronic. It can be a polycistronic message, which means that this mRNA codes for proteins, which can be from multiple genes in that bacterial DNA. So different proteins can be produced. When this mRNA goes to the ribosomes can produce multiple kinds of proteins from that one mRNA. This is not possible in eukaryotes. It's possible only in prokaryotes. And that is what polycistronic means. Second, are there enhancers in prokaryotes? I don't know. I don't think so. Actually, there might be. There might be. You never know. But in this lecture, it wasn't mentioned, and it wasn't mentioned on the sources that I checked either. However, you know, you never know. Enhancers might exist, but I doubt they're going to ask you that. They're not going to ask you. But you should know what an enhancer is. Like, you should know the concept. You should understand it. If someone asks you what is an enhancer, you should be able to explain it. Yeah. All right, someone else is asking, can you please tell us about the Shine-Dalgarno sequence? So the Shine-Dalgarno sequence, this is more important for translation. It's really a portion of the RNA in prokaryotes, the mRNA that is produced. It's a portion of the mRNA which indicates to the ribosome where to start translation from. So the Shine-Dalgarno sequence is it's essentially just for the ribosome it's a ribosomal binding site basically. So it brings the ribosome to the mRNA, and that point is where the ribosome is then going to start protein synthesis. So it has a lot to do with aligning, and like it brings the start codon towards the ribosome. I haven't covered translation in this lecture, so it's probably better if you go over translation. I think you have a different lecture on that. So it would be covered there, inshallah. It would make more sense to you because right now I don't have diagrams to explain it. But essentially, it's really just that one part of the mRNA which signals to the ribosome, oh, here's where you start protein synthesis. Cool. Everyone, everything clear? Yeah. Anybody else have any more questions? All right, that's it. I'm done. You know, good luck in your exam tomorrow. I know it's at like 8 a.m. or something. Inshallah, you guys will do great. And I think you have a part 2 now. I'm not sure, but I shall leave. Can I leave? Where's the host? Okay, all right. All right, thank you. See you guys.